We are going to be discussing some current SFC applications, some relatively new technologies that have been introduced, and how those can have an impact for us in the pharma and biopharma fields. While this is in no way intended to be a theory course on SFC, but rather a forum to talk about new and exciting applications, I do want to take a moment and consider what makes SFC special and adds a dimension to it as a mode of chromatography that is different from traditional LC methods. And primarily that comes in the form of the ability to control the density of the solvent directly through the use of the back pressure regulator. Think of it as adding an additional knob to what we get in terms of GC and LC. If when we move from GC to LC, we go from just changing temperature to being able to change temperature and mobile phase, we add on that dimension of pressure and density control when we go to SFC mode. Another inherent benefit of using SFC as a technique is the ability to increase linear velocity or mobile phase flow rate, and thereby shorten analysis period or purification period without losing theoretical plate count from our columns. So we can see here that in theory and in practice, we can have a much wider flow rate range with SFC or UHPLC columns than we can with either traditional HPLC or UHPLC columns that have a very narrow nadir to their Van Deventer plot, indicating an ideal flow rate, oftentimes that is much lower than we might like in order to speed up our analysis. And perhaps the most compelling reason to look to SFC is to look to alternative selectivity. The plot you're seeing in front of you constitutes a number of green and gray dots. The gray dots represent a series of compounds that are being separated under reverse phase conditions. And we can see a clear trend line in those that relates their percent retention directly to their log P value. The same compounds are represented by the green dots, only being separated under SFC conditions. In both cases, the stationary phase is a USP-L1 phase. And we can see that when we're considering reverse phase conditions, the only two values I need to know about two different compounds is their log P, and I can make a very strong prediction on when and where those will elute relative to one another. When we look at the spread of retention percent versus log P values, we find that substances, even with very similar log P values, can experience very different retention under SFC conditions, making it a much more versatile way to use even existing stationary phases. In an effort to make SFE and SFC more accessible to a wider range of users, Shimadzu has introduced a number of innovative systems, from our ultra-low volume back pressure regulator to our exclusive online SFE-SFC system for analytical coupling with mass spectrometry, and our compact benchtop semi-prep systems for purification. For the purification suite, we've designed a full range of instruments that are capable of micro-prep up to 150 ml per minute semi-prep operation, all centered around our revolutionary Lotus Stream gas liquid separator running under our LabSolutions software, dedicated software designed for and with pharmaceutical companies. In the area of software, Shimadzu has made an important innovation by including the SFC systems in both our method scouting and LabSolutions method development platforms. We also have compatibility for our SFC systems in walk-up user environments using our Open Solutions platform, which allows for the use of single quad and triple quad mass spectrometry as well. A wide range of analytical and prep SFC systems supports the use of all of our LC-MS detector types and is available in flexible configurations, including the Nexera UC hybrid UHPLC and SFC switching system. This system can be used in conjunction with method development software to support automated switching between SFC and LC mode and automated screening of up to 12 columns and multiple mobile phases, including use of the solvent blending feature to create custom proportion dissolvents on the fly without the need for premixing. Beginning with the introduction of our analytical system in 2014 and continuing for the last decade, Shimadzu has consistently introduced innovations in the SFC space to expand our SFC lineup and address the needs of SFC users in our community. With that, let's take a look at our first SFC application. This is going to be a case study of using our LabSolutions MD AQbD method development software to create and optimize SFC methods for peptide therapeutics. The method development process we use for SFC is going to be very much akin to the method development process for LC. We'll have a number of different mobile phase conditions we wish to screen against a number of different stationary phases in order to come up with an acceptable separation. We'll then interrogate that separation further by optimizing gradient conditions or other aspects of detection in order to come to a final method. When beginning a new method development task, selecting the correct columns to screen is always important. Within the SFC realm, we have access to columns as varied as normal phase columns, reverse phase columns, phenyl columns, chiral phases, almost anything that can be used under reverse phase or normal phase can be used under SFC conditions. This allows us to get incredibly varied separation behavior depending on the column chemistry we choose relative to our molecule. That's why there is the need to do screening. We saw earlier that you can get a relatively predictable separation relating to polarity by doing reverse phase. Remember that plot of the green and gray dots. There is no such model, though, as the hydrophobic subtraction model, if you've heard of that for chromatography, that relates directly to supercritical fluid chromatography. That means that you also have the advantage of being able to use a very, very wide array of these phases, but screening becomes an important task. Hence, you see the construction of systems often with multiple column support and automated column switching. To correlate with the method development software, a 6-column set was chosen for the development of the peptide separation methods. This included a diol column, a silica column, a poly-VP, which is a pyridynyl moiety on a polyvinyl extension from the support phase, the poly-BT, which is an aromatic separation column, the UCPBR, which is a perbromo-phenyl column, and an octadecyl silane column, a traditional C18. So a wide variety of phases ranging from traditional normal phase through C18. When choosing mobile phase systems as part of SFC method development, it's important to bear in mind that additives to the modifier can have a large effect even when only a small amount is added. In this case, we're examining the effects of water and ammonium hydroxide on the separation of ritonavir. On the left, we can see the effect of retention being lengthened and peak shape being slightly modified by the inclusion of water in the mobile phase. Even at as little as 5%, we can see a great shift in the retention time. And likewise, on the right, we examine ritonavir separation without water and ammonium hydroxide, and then the addition of only 0.15% ammonium hydroxide can again shift that retention time later, giving us the ability to tune separations to suit our needs and to generate retention with the use of these additives. So ultimately, our LabSolutions MD method development system is comprised of method scouting system hardware in conjunction with the method development support software, LabSolutions MD, and our columns that we've selected for our specific application, in this case, peptide separations. By combining these three elements together, we can streamline the SFC method development process by combining the systems, the columns, and the software into a unified working platform that allows us to have a singular goal. Incorporating all of those elements, we can now visualize our method development workflow for peptides as the screening phase and optimization phase, each subdivided into two sets of experiments. In the first experiment of the screening phase, we'll perform column screening, looking at our model sample, which is a mixture of commercially available peptides, including lopinavir, ritonavir, angiotensin 1, angiotensin 2, insulin, daptomycin, and pneumocandin. The second phase of the screening will be to look at acidic and basic additives, including formic acid, acetic acid, trifluoroacetic acid, ammonium formate, ammonium acetate, and ammonium trifluoroacetate. In the optimization phase, the first set of experiments will look at the effect of water addition and column temperature. We'll evaluate conditions of water inclusion from 0% up to 5% and column temperature in the range of 25 to 65 degrees for a total of 18 tested conditions. The second phase of optimization will take the information from the effect of water and column temperature and explore additional water ratios and gradient conditions to find the optimal conditions. In order to implement these experimental cases, we use the software aids built into LabSolutions method development. This allows us to easily select our modifiers, columns, and put in simple sample information that then automatically creates the batch schedule for analysis, including all required switching of mobile phases, equilibration of the system and of the columns, and all other considerations needed for valve switching, modifier switching, automating the process to remove the possibility of human error in the creation of multiple method files. For the column screening experiments, a 5% to 75% gradient running at 2.5 mils per minute was used with 0.1% formic acid in methanol as the modifier. Each of the included samples was present at approximately 1 milligram per milliliter and dissolved in DMSO for injection. The results of the column screening showed that the polar group columns, the poly-VP, SIL2, and diol-2 columns provided the best separation, with the poly-VP providing the highest resolution and greatest number of resolved peaks. The next set of experiments for additive screening then is informed by the choice of the poly-VP column, and the same 5% to 75% gradient at 2.5 mils per minute is run against that column for the 6 different additives. Results of the acidic and basic additive screening showed a preference for acidic additives with acetic acid and TFA showing nearly equivalent performance. Therefore, for the next investigation, both additives would be examined for the purposes of optimization, given that acetic acid would be a desirable choice versus TFA for the utility of using the system for multiple applications. So at this point, for now, minimal effort for configuring the screening of both our columns and our mobile phases, we now have a suitable result to move on to the optimization phase. As we move, we will be trying 2 different mobile phase conditions, methanol and acetic acid, or methanol and TFA, along with the Shimpack QC poly-VP column. For both of the acidic additives, experiments were carried out in a similar manner, testing concentrations from 0 to 5% water against column temperatures 25, 45, and 65 degrees, leading to 18 conditions for each additive being tested. In this slide, we can see acetic acid results, and on the left at the bottom, we see the result from 0% water and a low column temperature of 25 degrees. In this case, resolution between angiotensin 1 and 2 is not able to be achieved, and peak shapes generally suffer as well as having later elution. When we step up to 3% water, we can see that angiotensin 1 and 2 begin to become resolved, even at 25 degrees C. But by the time we've increased that temperature to 45, on the right, we can see we're nearly to baseline resolution of the 2 angiotensins. Here is a summary of conditions for the water ratio and column temperature experiments. And again, the screening gradient from 5 to 75% was used at 2.5 mils per minute of total flow. Looking in greater detail at the design space analysis for the acetic acid results, we can see that the lower column temperatures did indeed provide better results, with 25 degrees C identified as the best choice. And we see that adding additional water concentration increases the resolution of angiotensin 1 and 2. However, the overall performance of acetic acid as an additive still demonstrates relatively poor peak shape, with leading or tailing noticed on most of the peaks assayed. Owing to the relatively poor performance of acetic acid as the acidic modifier when subjected to further optimization, the choice was made to do gradient optimization only with TFA, with gradient endpoint being set at 55%, 65%, and 75% to induce a different slope and potentially alternative separation. Looking at the results from the design space of TFA in the water and column temperature stage of investigation, we can see that again, the lower temperatures, 25 degrees C, provided the best result. And if we look at the color heat map in the middle of the slide, we can see the red area in the upper left corner. This is oftentimes an indication that we may have additional design space that needs to be explored. We can see that in terms of the overall separation, the peak shapes are much better than those observed with acetic acid. Because that area of most favorable conditions was found at the edge of our design space, it does suggest that furthering those parameters may increase the available separation space. In this case, the suggestion would be either to go to lower column temperatures or higher concentrations of water. Because we're already at 25 degrees, which is the low end for doing SFC work, we don't necessarily have a lot of lower that we can go in terms of column temperature, as it may affect phase composition, but we can indeed investigate higher concentrations of water. Upon completing the extended gradient optimization and increased water concentration experiments, the design space revealed that increasing water was only effective to one further point, and that beyond 6% water, no additional gains in resolution were observed. In terms of gradient final concentration and slope of the gradient, very little effect on resolution was observed, with each being an equivalent choice. So let's shift gears now from an extended case study of developing a method for peptides to an application brief where we look at the exposed polar surface area assay for analytical SFC. Exposed polar surface area testing, or EPSA testing, is an assessment tool used for cyclic peptide cell uptake equivalency and is intended to replace or at least predict the results of CACO2 testing for cell permeability. What this does is allows what is typically a 5 to 12 hour uptake test to be accurately predicted by a 5-minute supercritical fluid or enhanced fluidity LC method. EPSA analysis uses a specific chiral selector column along with common pharmaceutical compounds to generate retention times for those compounds, which are assigned an EPSA equivalence value. Once the regression is created between those retention times, the EPSA score or value can be assessed for any cyclic peptide that is injected under the same conditions. The original methodologies for EPSA were approximately 15-minute methods. In 2018, Romulo Romero and company showed that they were able to shorten that effectively to a 5-minute run, and Shimadzu then looked at extending those modifier gradients to increase the speed of EPSA, and were able to find that the gradient could be sped up while still maintaining the elution order and maintaining acceptable linearity of the elution time regression. And despite the fact that we've mentioned before that chiral applications are not the only applications for SFC, we would certainly be remiss in ignoring the fact that that is one of the excellent spaces to use SFC. So let's quickly look at some innovations related to chiral columns and how the application space is being adapted. While we've mentioned many times that the variety of columns available for SFC separations is certainly wide, one of the things that was late in the game coming to SFC was the advent of small particle columns. So traditional columns, 4.6 millimeter inner diameter, 250 millimeter length, 5 micron particles are typical in chiral separations. And we can see here that we have a relatively rapid, less than 4-minute separation that we can achieve using a traditional format column, because we do have the ability to run at relatively high flow rates. As we though move to a 1.6 micron particle column, we see that we can actually get an equivalent separation, or at least a baseline resolution separation, of those two chiral compounds in less than 12 seconds. And by having the ability with the Shimadzu system to go up to 660 bar, we're able to go from a 4-minute run to a 12-second run. Even when comparing separations on 1.6 micron stationary phases, we can see large differences from system to system. In the example chromatogram shown, we're running on a system that's limited to a maximum pressure of 400 bar for the pumps. If we instead look at moving to the 660 bar system, we can increase that flow rate from 1.5 mils per minute up to 3.4 mils per minute and achieve a 50% reduction in runtime. Within the world of lipidomics, we can also find situations where naturally occurring triglycerides contain both enantiomers and positional isomers. These separations are rife for the use of chiral columns as well to optimize separation. So we've mentioned how we have the extra knob of back pressure in SFC versus LC or GC. So let's see how that's actually going to affect real separation. In this case, we have the 6 different species of positional isomers and enantiomers of our tags. When we adjust the temperature, we can see the expected trend of increased temperature leading to earlier elution. However, we can see that that's not really increasing our resolution between any of the species. And in fact, at 20 degrees C, while it has the longest elution time, we see potentially the best separation. 30 degrees is a nice compromise. We haven't lost much resolution. We haven't gained any either, but we've shortened the entire analysis time. Modifier concentration, at 20%, we see the widest spread of our peaks, not unexpected, as we're allowing more interaction with the stationary phase rather than partitioning into the mobile phase. 30% and 40%, we're not seeing any changes in selectivity, just earlier elution and less resolution between each of the peaks themselves. However, as we look at back pressure, at 10 megapascal, we see really no influence. That is kind of what we used as our screening condition. At 15 megapascal, we're starting to notice an interesting effect. Our species with a single saturation are eluting later, the pink trace, whereas our species with 2 unsaturated legs to the tag are moving to earlier elution. And as we increase the back pressure to 20 megapascals, we can see that effect increase. So we are actually inducing earlier elution of our early eluting compounds and later elution. In other words, increased selectivity by increasing our back pressure for these compounds. Putting that all together gives us a final LC-MS result showing our separation at 30 degrees C for the oven, modifier at 20%, and back pressure at 20 megapascal. So you can see that with just a small tweak to some of those parameters, we can increase that resolution on column. As our last set of applications to consider, I'd like to look at some new and emerging applications, particularly around oligonucleotides, microprep, and how we think about SFC and what enhanced fluidity really means. When we think about the workflow for synthesized oligonucleotides, we have the ability of creating those synthesized nucleotides with either a protecting group attached or without. By providing the protecting group, we can often achieve greater stability over time, less lability in process, but we add an additional step to the purification of those synthesized oligonucleotides, meaning that we have to first perform a purification, then deprotect, and purify again. If we can achieve separation, though, without having the protected group on at the initial stage, then we can reduce down the amount of time and effort required for oligonucleotide synthesis and purification. As seen here, the 10-mer separations conducted in SFC conditions were nearly equivalent whether the oligos were protected or not. This may allow for certain oligos to have that two-step in the purification process be eliminated by separating directly in either the protected or deprotected mode. As mentioned previously, the Lotus Stream gas liquid separator was a major innovation Shimadzu designed for the release of our PrEP SFC system. And the original version is able to handle the range from 150 mils per minute down to as low as 3 mils per minute. Its elegant design incorporates no moving parts and can be easily cleaned by simply submerging the entire gas liquid separator in an appropriate solvent and using the sonicator. The recently released Micro Lotus Stream gas liquid separator now brings the same performance and ease of maintenance to analytical fraction collection and is a first in the industry to be coupled with SFC. This allows the useful range to be extended down to as low as 1 mil per minute and up to 5 mils per minute using the Micro Lotus Stream. It's easily swapped in the fraction collector for the standard gas liquid separator and has the ability to collect into 96 well plates or any other format, included uncapped vials as seen here. So where is the future of SFC and enhanced fluidity chromatography? The sky is potentially the limit. With recent things entering the literature, such as the dual gradient UC ternary gradient shown here, we can think about separations that run the entire range of polarity, starting at SFC conditions and finishing at reverse phase aqueous conditions, allowing for the separation of proteins, here in this case showing cytochrome C, myoglobin, lysozyme, bovine albumin, and human transferrin. And this same technique has been demonstrated for proteins up to 160 kilodalton. We need to start thinking of SFC and the inclusion of CO2 mobile phases as part of a continuum of chromatography. From academic researchers like Susan Olesic, who popularized the term enhanced fluidity chromatography, to the godfather of SFC, Terry Berger, we should be thinking about this less in terms of, are we in the supercritical state? Because supercriticality may not be the real access point for using CO2 as part of our chromatography scheme. As Terry Berger once said, if he had to do it again, he would have avoided the term supercritical fluid chromatography being attached to the use of liquid CO2. And instead, he preferred the term compressible fluid chromatography. And I think that's what we need to think about. Chromatography is based first and foremost on repeatability. And if we can create a homogeneous mobile phase to use against our stationary phases, we can often achieve that repeatability. Whether or not we've technically gone in our triple point diagram into that supercritical space may be irrelevant if it supplies us useful separations. As we wrap up our event today, I hope that this has been an exercise in stimulating everyone's imagination for the possibilities of where SFC techniques and ideas can take us. Hopefully, we've provided you with an overview of the emerging hardware and software technology that continues to expand the utility of this technique for challenging separations. We've also been able to see how the use of automation software allows for the investigation of multiple mobile phases and multiple stationary phases with great ease for the end user, and how we can use that to extend the technique that we normally think of for small molecules and chiral substances into the realm of biomolecules, peptides, oligonucleotides, and even proteins. We know that SFC is always a great utility for separating chiral compounds, for doing purification with a minimum of dry down, a minimum of used solvent. But we should also be thinking how these techniques can be integrated with things like mass spec detection, and how we can apply that in areas like ADME and DMPK to gain what we can from the enhanced sensitivity as well as chiral selectivity of SFC. So I hope that this has been insightful to you and that you can find uses for SFC techniques within your own quests in pharma and biopharma. Again, I want to thank everyone for your attention today, and thank you for coming to our live event. I'm going to hand it over here in just a moment to my colleague Kevin for our live Q&A session. Thank you again.